Welcome back to the nervous tissue part 2. In the first part of this recorded lecture, I presented all of the chemical and cellular background that sets us up for understanding a nerve impulse. The steady state, which is resting potential and graded potentials, were also described. So what does that mean with respect to the nerve impulse? We're now ready to fire a nerve. And that means reaching the action potential, because graded potentials are small force perturbations that cover the membrane of the cell at all times. They don't produce a nerve signal that propagates itself over the whole membrane of the nerve. How do we actually do that? Reach that threshold, the action potential, and propagate that nerve impulse. Well, here is the starting point. And to understand it, all we have to see are a couple of different gates. Again, orange showing potassium and purple showing sodium. Now, I want to make a point. We haven't drawn in the slow leak channels of those two ions that we drew earlier. We didn't draw in the sodium-potassium pump. They're still there, and they're still operating. But to understand the resting potential to action potential and the nervous impulse, we don't have to look at those. We just have to look at these special gates. These voltage-gated potassium channels and voltage-gated sodium channels that are going to produce the real movement of the ions. Now you're going to notice lots of sodium outside. Look at the purple. And lots of potassium inside. The negatives remind us of the overwhelming load of protein inside the cell and how that contributes to the negative charge of the cell, the resting potential. The first thing that occurs is a graded potential, whether it's due to a physical or chemical or electrical stimulation, reaches a particular voltage value. We're going to choose minus 60 millivolts as that value where the graded potential hits threshold. Now, the importance of that value is that's the value where the sodium gate pops open. Now, you'll recall the big arrow for sodium at rest, pushing it into the cell. So when that pops open, sodium is going to flood into the cell, and the voltage is going to change from minus 60 to minus 50, 40, 30, all the way past zero, and we're now observing it a little bit later when it swung from minus 60 all the way over to plus 10 millivolts from the movement of sodium. However, as sodium rushes across, the concentration on both sides of the membrane becomes closer and closer to equal, and so the force of diffusion goes way down. Also, as we cross zero and become positive, the electrical force which drew sodium in at the beginning has now been eliminated and even reversed. So the force on sodium to move after this initial burst is much reduced. But at minus 10 millivolts, then rapid depolarization, another thing happens. When you reach plus 30 millivolts, and you'll notice plus 30 millivolts causes another change right here. It closes the sodium channel, so sodium can no longer move, and it pops open the potassium channel. Now, potassium is very concentrated in the cell, which is now positive in its charge. So if you open a gate, the electrical effect is to push potassium out. Also, it's very concentrated in the cell and very dilute outside the cell, so the physical force of diffusion is to push potassium out. So at plus 30, you would have depolarized from minus 60 all the way to plus 30. Now you're going to pop these gates in the opposite direction, and potassium will move, and that needle is going to swing back toward the resting potential. So as the potassium moves across, in response to those chemical and electrical forces, the needle begins to swing, and it goes a long way. The negative charge that we're reestablishing pushes the potassium harder and harder, and eventually you end up with a lot of potassium outside the cell. Now, sodium cannot move. Its channel is closed. But the overall forces balance out, and potassium keeps moving until you reach this negative value. You'll notice that it's not just repolarized, it's not minus 70, it's taken past minus 70 to what we call a hyperpolarized condition. At that point, the potassium gate slams closed. Now at that point, the electrical impulse has basically spread from this point in all directions and is popping sodium first, then potassium, sodium first, then potassium in ever increasing rings in every direction. So here's what it looks like on a voltage chart. Over here we again have voltage. This is the 0 mark, and this is the resting potential. We have here, below threshold, anything that happens below that dotted line would be called a graded potential. And it could basically move a lot until it hits that dotted line. When it hits that dotted line and the sodium channels open, it becomes much more positive very quickly. This is called depolarization. And then the sodium gates close and potassium opens, and potassium moves in the opposite direction. Potassium moves repolarizing and hyperpolarizing until all the channels close. Now at that point, you notice we're below the resting potential, but this is where the slow leak channels allow the movement back to the sodium outside, potassium inside configuration, and the sodium-potassium pump is burning ATP and can burn ATP quickly to reestablish that resting potential. So this kind of glitch, this kind of electrical tracing is indicative of the nerve impulse that we generated by this action potential. So to compare, we've already talked about the characteristics of graded potential. How do action potentials differ? First of all, you reach threshold and pop that sodium gate, it's always going to depolarize. It's not going to hyperpolarize. The depolarization to threshold has to occur before the action potential begins. There is a threshold value. It's all or none. Once you reach the action potential on one portion of the membrane, it is a strong enough effect that it pops the sodium gates in ever widening circles around the entire body of the cell and down the axon to the telodendria. It will reach the telodendria and their vesicles with neurotransmitter. The action potential at one site will depolarize adjacent sites to threshold. So it is a self-propagating rather than a limited passive stimulus. The entire membrane is depolarized, but one difference between graded and potentials, there's a constant variation in graded potentials. They can go up and down almost indefinitely. Action potential, after you fire the nerve, there's a recovery period. Close the gates, shove the sodium out, bring the potassium in, and you restore a sensitivity. That nerve is then ready to fire again. The graded potentials are occurring on almost all plasma membranes in the body on a constant basis. But action potentials are characteristic of what we call excitable membranes, sensitive membranes, which means you poke them and they do something. We've now got two examples of those, the neurons of the nervous system, which propagate signals for fast signaling, and the muscle cells that produce contraction and movement. Now, we can see this model. I apologize that this is so small. It's kind of good it's on your computer screen. Maybe you can enlarge it. But you'll notice that that pattern of an action potential, which then basically moves from gate to gate all the way down this axon. So you can see depolarized, intermediate, and resting, moving in this direction as the sodium rushes in and the potassium is pushed out. This is called continuous propagation, and it's characteristic of axons that have no coverings. So it is almost as if you have to walk along here and pop every single sodium gate in order to get the electrical impulse to move. Now, naked neurons are transmit at a fairly rapid rate, and they constitute what is called gray matter in the nervous system. In the brain and spinal cord and in the ganglia, you can see a difference between what we call gray matter and what we call white matter. And I want to explain that difference because it has an effect on the speed at which the neuron conducts the impulse. This shows an axon down here that has been covered by those myelin sheaths of the Schwann cells. Do you recall that they were like beads on a string, and they covered the membrane here, and they left a small bit of the membrane exposed, one that could be stimulated from the outside. Now, a curious thing happens when you myelinate, like up here. These are what we see in peripheral nerves, these myelinated axons covered with Schwann cells. When you do that, you end up with nodes that are excitable and a whole long length that is insulated. So here we have the first action potential in figure 2 with sodium rushing in. But basically that sodium has to jump over, sort of like leapfrogging this insulated region, and can only find the next gate here at the node. And so as a result, instead of tripping every sodium gate along this length, it basically leaps over and goes to node 2. Node 2 is flooded with sodium and it leaps to node 3. So what I'm saying about this myelinated axon is that insulating these segments with myelin sheath actually speeds up the transmission of an electrical impulse. It makes the electrical impulse move through it more quickly. When we have myelinated axons, they form what's called white matter in the brain, spinal cord, and the peripheral and the ganglia, but also in the peripheral nerves. Think of these as transmission lines. Think about a heat sensor here in the finger. When I put it down on a burning skillet, on a hot skillet, it's going to destroy cells. Now that's not normally something I have to think about or decide. I want that signal from that sensor to move to the central nervous system as fast as possible. And going through the arm, it doesn't have to stop and tell the wrist and tell the elbow what's happening. You basically are going to want to move that off. That's going to produce a reflex jerk response that goes and moves your finger off. White matter is what forms those very fastest transmission lines and what connects the centers of gray matter throughout the nervous system with communication among regions. Now, we have delivered then the electrical impulse, I think pretty efficiently, to this structure, the telodendron, the presynaptic membrane, and the synapse. The synapse is that gap, and we see it here as we saw it with the neuromuscular junction separating the presynaptic membrane from the postsynaptic membrane. The pattern is the same. Number one, here comes the electrical impulse. When it arrives, I got excited. When it arrives, gates in the presynaptic membrane open and calcium rushes in. Kind of odd. Calcium is what unlocked the sarcomeres. Calcium is going to be what causes the fusion of vesicles with neurotransmitter with the presynaptic membrane, number two, and the transfer of acetylcholine into the synaptic cleft where it binds receptors on the postsynaptic membrane. That's the same pattern we saw before. Enough binding and sodium gates pop open. Now we saw that sodium gates were what the response to an action potential on the original action potential sequence that we described. And that's going to start the action potential on the postsynaptic cell. Now, the acetylcholine is stripped off as before and decomposed. Some of that will be thrown away as waste. Other parts of it will be transported back. You can see the choline, which is big and hard to make, goes right back in where acetyl-CoA from the mitochondria, our familiar 2-carbon fragment, combines with it and makes acetylcholine again and packages it in vesicles. That's part of the recovery period when the nerve is insensitive and preparing to fire again. The mechanism for neurotransmitter function is not limited to acetylcholine. It was the first one we discovered, and it's by far our best known. But when we look through the body, we see a number of chemicals that are natural to us that have neurotransmitter substances. When we start playing with drugs, we notice another set of chemicals that produce neurotransmitter effects. So up at the top, acetylcholine is a great example of a direct effect on a chemical gate. Also, the amino acids glutamate and aspartate are direct neurotransmitters. You bind the site, it pops open. There are also a number of indirect effects. You can see the examples listed up there. They stand for the hormones epinephrine and norepinephrine, dopamine, histamine, and a carrier I'll just call GABA. The idea here is the neurotransmitter binds a protein that starts a biochemical cascade inside the cell. The result is cyclic AMP, which typically appears when you have low energy or when you need energy at the maximum amount. Cyclic AMP then will open the channel. That's the mechanism illustrated in B. In C, indirect effects via intracellular enzyme, it's known that a couple of very small but important molecules can have neurotransmitter effects. They diffuse directly through the membrane. Nitric oxide and carbon monoxide both have this effect once inside the cell. They activate secondary messengers that open ion channels and also change the cell metabolism and activity in important ways. So neurotransmitters are important, and understanding neurotransmitters is important to our medicine and our chemical drug problems. So a couple of things to review and underline about nervous tissue. We're talking here about sensitive membranes, again, just like we did with the fiber of a muscle. The action potential, which is the threshold which fires the nerve, and the necessity for a synaptic transfer of that energy by a chemical step. And that basically explains nerve impulses within the individual cell. But just like the muscle, once we understood the sarcomere, we had to add that sarcomere action up to figure out how does the muscle actually pull in its ends. And these neurons are arranged in networks which are much more complicated than simple electric circuits. You know, electric circuits, if you have a break in the insulation, or if you connect part of the circuit to the ground, it diverts the energy away from the appliance. So the light bulb doesn't light or the toaster doesn't toast. Whereas the characteristic of these nerve networks is that each neuron is connected off to hundreds or thousands of other neurons. And as you get into denser and denser collection of neurons, the number of synapses per cell goes up to tens of thousands or even hundreds of thousands. So what does that add up to when we look at something like the brain? Well, that adds up to a pattern of electrical discharge that goes over the surface of the brain and is developed as a combination of your growth and your genetics and the experiences that you incorporate in that brain. So this pattern of electrical discharges, called electrocardiograms or ECGs, reflect how the firing of the nerve networks you've developed add up in your brain. At some point you make the transition between simple signals here. So it's easy to understand what we're doing with a reflex action to a burn. We're basically preserving cells. It's a little more difficult to see how that process adds up to things like memory, like learning, like personality, or things like skill memories. When you're a baby, you can't stand and walk, but you learn to do that, and then it becomes automatic. It is voluntary, whether I stand up or not, but I basically am able to stand and maintain my balance and move based on my familiarity with the balance and movement. We would call them feelings that I have when I'm doing that activity. That's a skill memory. How do those synapses and axons add up to that? We're going to look at that in detail in a later chapter. Let's just look on the single neuron level and figure out how do we reach an action potential, because each nerve impulse that arrives at a synapse doesn't necessarily fire the next nerve. There are two main ways that you can get graded potentials to add up to an action potential. This first one is called temporal. And the idea is this one synapse is going to send a message down and produce this graded potential, but before it is completely dispersed, a second stimulus arrives just a little bit later. The change that it produces hits the action potential, and the propagation of the action potential move down the axon. The idea with temporal is multiple rapid signals down the same track. The obvious other change is shown here, where you have two synapses that are grounded or established on the body of the neuron close together. So even though there are small impulses arriving, they add up. This is like two pebbles landing close to each other in the pond. The wave that produces an action potential, and it propagates down the axon. So the whole idea here is small contributions, small graded potentials have ways of summing up over time or over space on the body of a neuron. We would represent it this way based on whether these are excitatory, are we depolarizing, or inhibitory responses that are produced by different stimuli. And in fact, looking at this, the resting potential doesn't really change if excitatory and inhibitory are applied at the same time. We are seeing that the built-in complexity is something that we're just beginning to understand. Synapses between the axons of two neurons can be affected by a third neuron that has a separate synapse on the presynaptic membrane. So although the main track may produce a straight line stimulation, this third axon can deliver a signal, either electrical or chemical, that will either inhibit the firing of that synapse or will amplify the firing. And let's take a look at what we mean by that. Here's a main track of the tan to the purple cell, and a third neuron, which is a neuromodulator, one that's going to affect the transmission of impulses from tan to purple. In this first example, number 1, see the action potential arrives and calcium gates pop open, but an action potential arrives from the neuromodulator and releases GABA. Now the effect of GABA is that it inactivates or it blocks these calcium channels. So this is an example of presynaptic inhibition. If we block these channels, less calcium enters, less neurotransmitter is released, and we reduce the effect on the postsynaptic membrane. So we call this here presynaptic inhibition. In this second example, same diagram and idea. Instead of GABA, we release serotonin. Serotonin activates the calcium channels, so the opposite effect, more calcium, more neurotransmitter, and an increased effect. We would call this effect here presynaptic facilitation. In fact, this is as far as we can go with simple cellular and chemical models. It in fact is a very, very rapid system that is firing nerve impulses and then spreading those nerve impulses out in waves through nerve networks to larger and larger associations of neurons. The frequency of action potential depends on the degree of depolarization above threshold. Holding membrane above threshold level has the same effect as a second larger stimulus and reduces the rest or recovery period. In the nervous system, it's the change in the transmembrane potential in a very short period of time that will determine whether action potentials are reached and are generated. That is the simplest form of information processing. So in summary, we're going to use this fast signaling system to transmit electrical signals. It's going to depend on the strength of the stimulus and the graded potential adding up to action potentials. Whether those action potentials add up in a spatial or temporal way to propagate across the synapse and into the presynaptic, into the postsynaptic circuit is really the determining factor of whether the nerve impulse goes on or whether it stops. This idea of a neuromodulator. A neuromodulator is a third neuron affecting a synapse, which can either inhibit or facilitate the transmission of that signal across the synapse. So this gives a bit of reflection on the various psychoactive drugs that we have. You know that sometimes they are simple in their design, wishing to affect nerve signals that we interpret as pain. So we're trying to relieve pain. In other cases, drugs are used to create the activation of a higher order function of the brain. So for example, the experience of seeing, hearing, tasting, touching, smelling the world around us is a very real perception. But if we are generating an image like a dream, which is generating images that are not really there, we would call that a hallucination. And psychoactive drugs can produce those as well. So the summary information brings nervous tissue to a close. And thank you very much.